Hello, everyone. Welcome to the last webinar of the year. We're going to wait a few minutes to let everybody get into the room. In the meantime, why don't you all put in the chat where you're from? We'll see where everybody is from today. Odessa and Lakeland, Bellevue, Lake Placid, all over today. Everybody's from all over Gainesville, Tallahassee, Miami. We've got a big crowd today too. We'll wait one more minute. We'll wait one more minute and we'll let Dr. Schutzman speak. Santa Rosa, that's way out there, Santa Rosa County. In Port St. Lucie, St. John's. We've got a lot of people today from all over. Longwood and the Villages. Let's see, let's go ahead and start. Looks like people are in the room now. Welcome to the Florida Friendly Landscaping Program's Homeowner Webinar Series. It's a series given every third Tuesday of the month at 11 a.m. My name is Jen Marvin, and I'm the statewide education coordinator for Florida Friendly Landscaping. Today we have Dr. Bart Schutzman, who will speak to us about plant propagation. First, let me go over a couple of things. Your microphones have been muted. If you have questions, please type them into the Q&A box instead of the chat box, and we will answer them at the end of the presentation. Stick around also for the survey at the end of the presentation. It helps me give you the kind of programming that you like, and it lets us know how we're doing. Our next webinar will be in January. We'll talk to Mark Frank, who is a botanist and assistant collection manager at the University of Florida Herbarium. That's January 21st at 11 a.m. Eastern Time. And he will be giving us a talk on the houseplant renaissance. So he'll be talking about, basically he'll be talking about his houseplants, which I think is pretty neat for botanist and herbarium employee to talk about that. So let's talk about Dr. Schutzman. Today we will be talking to Dr. Bart Schutzman. He received his BS degree in plant science from the University of California at Davis in 1980, and his MS and PhD degrees in plant science from the University of Florida in 1982 and 1998, respectively. His field of expertise is plant taxonomy. His MS and PhD research was conducted on the systematics of the cycad genus Zamia in Mesoamerica. He did extensive field research in Mexico, discovered and named several species of Zamia and related cycads. He currently teaches plant identification courses in the Environmental Horticulture Department at the University of Florida, and has also taught residential horticulture and plant propagation. Dr. Schutzman is the Director of Publications for the Cycad Society and was editor of the Cycad newsletter from 2000 to 2017. Dr. Schutzman, thank you so much for joining us today, and please take it away.
Well, thank you, Jen. It's great to be here. I've been involved in propagation for a long time, but never really gave a talk on it. I teach propagation at UF, along with plant identification. And propagation is truthfully one of my favorite activities anyway, as I imagine it is for many of you who are watching this talk today, because plant propagation is fun because it gives you free plants. So what else do we need to say? But plant propagation is, it's an ancient activity that goes all the way back to when humankind first began to cultivate plants instead of to replace hunting and gathering. Now, of course, we didn't flip a light switch and it went from hunter-gatherer to farmer in one fell swoop. There's probably a good bit of transition involved. But in order to grow these crops, we had to select the ones we wanted, and we had to propagate them. So propagation is hand in hand with the beginnings of our modern civilization. And so that's why plant propagation is so important, and we emphasize it a lot in our department. So to create new plants from existing ones, when we do that, we also have the option of selecting the best ones. So when you grow whatever plant you're going to grow, and you grow maybe thousands of them from seeds, and you see that one has done much better than another, you can select that one to collect further seeds. So over time, we improve our seed batches and other plants. We propagate the ones that are most useful to us. And this is, again, not a new activity. This propagation goes all the way back to our earliest antiquity. So when we talk about propagation, a couple of terms. Well, propagule is one that we bandy around, and it refers to the part of the plant that we use to propagate. It's called a propagule. So that's more of a technical term, but you'll see it. We have to be concerned about those because that's the part we want, and we want to make a new plant from it. Then we have to be concerned with the environment that we are going to grow this propagule in to create a fully functional plant out of it. And number three, how we handle it to do that. So we've got to be concerned with all of these things. Now, I mentioned right away greenhouses because they are really very important in all types of sophisticated propagation schemes. Even a lot of times when plants are going to be put out in the field, they're not always direct seeded or planted from small sprigs directly into the field. A lot of times they are, most of the times they are grown up in greenhouses. And so I like to bring up a tiny little bit of history here just because it's fun. Greenhouses go back all the way to ancient Rome. That's our first known creation of a greenhouse. It may not be the first, but it's the first we know of. And if you've heard of the Emperor Tiberius, I think he was famous for other things like conquering and slaughtering his enemies or whatever. But well, we think of him in propagation because he was asked by his doctor to eat a fresh vegetable every day. And in order to do that, they basically had to create for him somewhat of a greenhouse. So he lived on the beautiful Isle of Capri and created there something called the specularium, which was kind of a greenhouse of sorts. It was basically a walled-in structure, and they didn't have sheet glass back then. So what they did is they peeled sheets off of mica and created a light transmitting ceiling and roof. And then a cart with the plants that they were going to grow was wheeled in and out of there, brought in during bad weather, so it still would get sunlight. And therefore, Emperor Tiberius got his daily dose of cucumbers. So, back in the ancient days of Korea, we know of greenhouses being heated by a special method called the Ondol method, and we know that this was done as early as the 1400s. In the US, the first known greenhouse was in the 1730s. And today, when you go to see greenhouses, you'll see a very large range of structures, very sophisticated. So what we've done, we haven't really added any huge amount of intelligence to our activities. What we have added is technology. And if you know what's basically the theory about human beings is that we've not really grown our craniums very much in the last several thousand years, but we have added significant amounts of technology, so we can do things that we never did before. So anyway, greenhouses are really foundational in the plant propagation activities. Now, when you get down and dirty in propagation, you realize right away that every plant is its own individual, and we shouldn't assume anything about how it might be propagated. Every plant is genetically different. The only exception to that is if we are cloning a plant that is a clone, and most of the time the genetics of clones are identical. There can be small mutations, but generally they're identical. And so when we know how to propagate a certain plant and it's a clone, we kind of know the best way to do it. And then groups of plants, different groups of plants, we know generally how to propagate them. So many people refer to propagation as both an art and a science. And this is when I'd like to mention that the reason we say that is because if you tried to scientifically manipulate every single variable concerned to find the best way to propagate a plant, you would be working on one plant basically forever because the number of variables are huge. And so therefore, the propagation methods kind of wander into the area of art because we are using our imaginations to come up with possible methods. And so we start back in antiquity, there really wasn't any scientific basis for any of what people were doing. They were using trial and error, and science was the afterthought. So right now, it's still done that way by most people. Now, the difference being you can go to some very good references, and I'll mention a few of them at the end, and find information about what people before us have done. And thankfully, they're recording their successes and failures so that we have starting points for many, many plants in the ways that we might propagate them. So as far as talking about science and art, back as far as 1660, this fellow Robert Sharrock wrote this book. It's the first we know of where propagation is specifically discussed. Now, when propagation is done commercially, we know that we can't come up with the perfect environment for each and every plant that's going to be propagated. So methods we come up with methods, I should say it that way, that meet the middle ground. In other words, if we get to the middle ground where most plants do well, if not at least okay, we can have a general environment to propagate plants and do pretty well with most things. Now, there are always exceptions to that, but the middle ground is where we aim for. And unless you have a very specific plant and you're going to be producing that forever, you can tailor your environment specifically for that plant. But I can say here at UF, we have something we call the mist house that has different settings for timing and the amount of mist that's emitted from little sprinklers. And we put flats of cuttings in there with the hopes that most things will root while they're in there. So we'll talk about that a little bit when I get to cuttings. So what types of propagation do we have? And I've listed it as 7 methods. We'll briefly go over them, starting with seed. This is probably most of you have done at least 2 or 3 of these methods. Most of us have planted seed before, but it may seem like a simple act, but there's a lot of variables in there that we look at, especially when we're not planting a seed that's easy to grow. Those are the ones that everyone has done. When you buy your vegetable seed packet at the store, you plant that. You don't really have to give it much thought other than watering it, and up comes the plant. Well, that's the easy part. But anyway, we'll get to that in a minute. Then we have cuttings, and anyone who has a grandma knows what a cutting is because she made them. And that's where we learned how to propagate a lot of things, because she had some great little plants. She made cuttings and distributed plants to her friends. And in the old days, cuttings were very important too, because not everything was easy to grow from seed. The third type I've listed here is division. And in that case, a plant gets to be quite large and forms a clump. In that case, we can either dig it up, or we can take a sharp knife or a machete or something like that, put it in the ground and chop that plant in 2 or 3 and make pieces of it. That's called division. The fourth one, not as many people have done, and there are many different types of it, but layering is a way to produce, we'll say, cuttings without removing them from the mother plant. So when you make a cutting, you're cutting the little branch or whatever you're taking off, you're removing it from the mother plant, and then you're trying to grow it. In the case of layering, we're doing things to the plant, to the branch of the plant or the piece that we want, to get it to produce roots and be able to be independent, but it is still connected to its mother plant. And the fifth method, which is it's fun to watch, a little harder to do, but I think it's doable with a little practice and skill, and that is grafting, where we take one type of a plant, a piece of it, and we connect it to another plant. The benefit of that is that the other plant may already be in the ground growing, and we're just altering what's on the top of that plant. So we'll talk a little more about that too. Budding, instead of taking a branch and putting it onto the other plant, we take just the bud, the ungrown bud that's found usually at the base of every leaf. You can remove the bud. It's very much done with citrus, and tuck it into the bark of the plant that you want it to grow on. And with some luck and skill, you'll have that plant growing. The last one that I mentioned here is probably the most recent, not probably, it is the most recent aspect of plant propagation. Really only been done for nearly 100 years, but much of that was the development of theory. And in this particular case, tissue culture involved a lot of science, because science was important during that period of time, and we needed it, because tissue culture is not something you automatically would come up with intuitively. And we'll talk about that. So when you look at the 7 different methods, you'll see I've got a big red X in the tissue culture line, and that's because it's really so difficult and expensive and requires so much training that the average homeowner or person that's casually going to do propagation would not really practically be able to conduct it. If I have time, I'll show you a couple of things about tissue culture, because I know it's a very interesting subject. When we go to tissue culture labs and see what they're doing, it's absolutely fascinating. But trying to do it at home is, for most intents and purposes, nearly impossible. Now, I've labeled cuttings with an asterisk, and down at the bottom I say some will work, some will not. Cuttings can be rather difficult in some cases. There are plants that are quite reticent to produce roots when you remove them from the mother plant, and we have to do special things to prop them up and make them survive long enough to produce new roots. So cuttings are not an automatic, yes, we can do them. They require quite a bit of effort in some cases to figure out just what method can be used to achieve success. So cuttings is kind of a maybe, but there are many, many, many plants that you don't really have to think about it too much, and some standard methods can get you results. Now, Jen was mentioning about the next webinar was going to talk about houseplants. Well, I'll tell you, cuttings are very commonly used to propagate those houseplants. So when Mark Frank talks to you about houseplants next time in January, you can be thinking, how can I propagate that plant? Dr. Schutzman said, mostly we can make cuttings. And that is essentially true. Now, as far as the tissue culture, also called micropropagation, I'm going to use an example. Because I've said things are rather impossible, but in the case of orchids, a very rare, expensive, and desirable plant in many cases, one had to wait for a long time until that plant was large enough to divide, where you could then take one plant and make possibly 2 or 3 plants out of it just by cutting it in separate pieces and planting each piece. That's the easy part. With propagation, by micropropagation or tissue culture, one plant could produce as many as you want just by continuing to proliferate it. And we'll talk about that at the end if I have time. But there is a reason why people like micropropagation, because you can start with one very good plant and produce as many as the market will bear. So that's why tissue culture is a big deal in commercial production. And here are some of the reasons why one wouldn't be doing tissue culture at home. It requires aseptic conditions, which truthfully, oftentimes they can't even get that done in a hospital, very sadly. And it requires that for tissue culture because when you take a tiny little piece of a plant and try to grow it into a full plant, it requires the absence of harmful fungi and bacteria, and that's hard to do. You need a lot of space. You need expensive technology, equipment, chemicals, control over temperature and humidity. And so for all these varied reasons, we don't do tissue culture at home. But if you're really excited about doing that and you have deep pockets, you know, I wouldn't doubt that people could get this done if they are driven and have the means to do it. So I don't want to tell anyone, oh, you can't do it, it's impossible, because it depends on your goals. So now let's go to the main ones. So we have seed propagation. And those of you who have planted seeds, it's a very fun operation. You punch a little seed down in the soil and you watch it grow. With all the technology we have now to study DNA and genetics, we found that the germination of a seed is not as simple as it looks. There was one study, I can't remember the details, but we could find out, where they determined that from the time that a seed first started to imbibe the water to begin the germination process and to germinate required the activities of some 3,000 genes over the period of time. So it's quite a complicated little set of circumstances that are going on in that seed. Whether it's easy for us or not, those things are happening. So other than just buying some seeds at the store that all you have to do is plant them and they come up, we have in seed propagation, we have what are called dormancies. There are different things that are built into seeds that prevent them from just automatically coming up. And I will tell you about those. So when you have seeds that have dormancies, you have to treat them first in order to plant them and then successfully get them to germinate. So one of the main things is the permeability of that seed to water, because water needs to reach the inside of the seed so that it can activate all the activities required for a seed to germinate. And when the seed has an impermeable coat, we have to what we call scarify that. And scarifying can be done by rubbing a seed on sandpaper or putting it in harsh acids, which requires some skill and a lot of care to do safely. But permeability of water is important, and that typically the impermeable seeds are ones that can be produced by the plant, land on the ground, and sit there for sometimes many, many years, and they're thoroughly protected by their hard seed coats uh, so that they can last longer, and then they can germinate when sufficient time has elapsed and natural forces can gradually reduce uh, the the or increase the permeability of the seed. So like if it's sitting, getting rained on, insects are trying to chew on it, uh, the weather is acting on it. It may be in the soil, and some fungi may try to eat on it. But over time, hard seed coats uh, get permeable enough so that enough water can get in, and then those seeds can germinate. So when we have seeds like that, we have to treat them ourselves to speed up that process. For the plant, that's a protection mechanism so that all of their seeds don't fall on the ground, grow, and then if something bad happens, there are no plants left um, uh, to germinate anymore. They've all germinated, so they might get wiped out. So hard seed coats are important evolutionary mechanism to protect those seeds. Another thing that can happen is that seeds can be released from their mother plant before the embryo is mature. So the embryo, the little embryo, the tiny little plant inside that seed must reach a certain maturity uh, before it can germinate. And sometimes we have to take seeds and store them, called stratification, uh, to let the embryo continue to grow, mature. Excuse me, not grow yet, but mature. Stratification can be done over a period of months, sometimes longer, but in some cases there are actually chemical treatments that can be applied to seeds to speed along that process. So there's a whole technology for overcoming that type of a dormancy, which is the embryo maturity. Then there's another one where maybe the seed can absorb water, and maybe the embryo is just fine, but there's a chemical in the seed that inhibits its growth. Why would it have that? Because the chemical is a certain way to achieve the same effect that you would have if you had a hard seed coat. In other words, a certain time would have to elapse where the seed would get rained on, or it might soak in some water for a while, and those chemicals have to leach out of the seed, and when they are sufficiently gone, then that seed can be activated and it can germinate. So in the cases of chemical inhibitors, a lot of times soaking of the seed is required to leach those chemicals out. So those are the three major types of dormancy. And these are not necessarily mutually exclusive. Uh, Sometimes one type of seed will have more than one dormancy and require multiple types of treatments in order to get it to grow. And I'll give you an example. Years ago, we tried to grow uh, Kyananthus, our native fringe tree, from seed, and it has a very complex series of protections. So the little fruits come on the fringe trees. They look like tiny little olives. So uh, the fleshy coating of those little seed that are in fruits, that fleshy coat, uh, serves as a chemical inhibitor. So those fleshy seed coats have to be removed, which if we want to grow that, we do that by hand, either by soaking them, using a knife, or scrubbing them, and we remove that fleshy coat. Once you've got the fleshy coat off of there, you have a hard seed coat that's fairly impermeable to water. So that's this first dormancy that I was talking about. That requires scarification, or in some cases, you don't have to use sandpaper or chemicals like acids. Sometimes you can just crack that, and that's what we did. We used a small pair of pliers and cracked the seed coat. That would allow water to get in. But guess what? There's still yet another problem with the seed of fringe tree, and that is the embryo is not mature when it's released. So how do we get by that? We treated those seeds after we cracked the seed coat. We soaked them in gibberellic acid solution, and that helps the embryo mature. Now, having gotten through all three dormancies, those seeds were then free to germinate, and germinate they did. But if you just tried to pull some seeds off a fringe tree and stick them in the ground, oh, they might germinate, but it might take a year or two before it would happen naturally, which is probably the whole reason why those seeds evolved that way, to make sure they didn't all just come up. So that's a very interesting aspect of seeds that not everyone will notice. If you're just planting vegetable gardens, you're not going to see that because all the seed for those vegetable gardens are ready, willing, and able to germinate when you buy them. In addition to the types of dormancies, we of course have to plant seeds in a proper media. So many seeds don't like to be in a flooded condition because that removes the oxygen that probably is needed for all the processes of germination, because oxygen is needed for almost everything living organisms do. And then whatever the soil you use has to have proper drainage, but at the same time, it also has to have enough moisture holding capacity to be moist enough to promote seed germination. So that's a consideration. Then, how deep and how do you orient the seed? That's an issue for some seeds. If you've ever seen, some seeds are shaped in such a way that one wonders, like say you took the seed out of a mango. Well, pretty much anywhere you planted it probably would eventually come up. But if you notice, the seed is very wide, but it's kind of flat. And would you plant it flat? Would you plant it the long way up and down? Or would you plant it sideways? And that the last answer is the one that I generally do, because when the seed coat cracks open, the little embryo sends out its shoot and comes straight out of that crack and goes straight out through the soil. So basically, by planting it in that orientation, it makes it easier for the plant to actually get above ground and start growing. So if you plant a seed too deep, if it's a small seed, it may not have enough energy for the seedling to grow past too much depth of soil. So that's, in some cases, an issue. And that's why you find that most tiny seeds are indicated to be put under a very thin layer of soil, because they theoretically don't have a lot of nutrition built into that tiny seed to allow it to do much else. It has to basically just come right out of the soil and grow, or else it's not going to be able to make it if you plant it at 3 or 4 inches deep. And then for the other parts of the environment, you might like, some seeds might like to have warmer conditions. So they have things like bottom heat mats, and you can plant them in trays with covers, which aids in preserving humidity. So those are kind of fun to do if you don't have a dedicated facility for seed germination. A lot of people resort to these starter kits and bottom heat, and they work quite well. If you've dealt at all with trying to grow succulents from seed. Succulents are very special plants. They come from very torrid, very warm areas, generally speaking, and that's when they germinate. So in most cases, succulent seeds have to be kept quite warm in order to grow. And sometimes that even involves temperatures as high as 75 or 80 degrees Fahrenheit. So one thing you may be noticing, I'm mentioning all these factors, is that it's important to do your homework when you're going to grow an unknown seed to see if anyone else has done it, to see if you can find enough information so that you don't have to reinvent the wheel. That's really important in plant propagation. And we'll talk more about that at the end. The next thing I want to mention, to me, one of the most fun things to do is to make cuttings. Cutting propagation is quite a fascinating activity because basically what you're doing is cutting a piece off of a plant. And that piece then, if it could talk and think, it would say, oh my God, I have no more, my roots are gone. How am I going to get water? How am I going to get moisture? And my God, I'm going to die. So no, we cut the piece off of the plant and we supply the proper conditions, and lo and behold, roots can emerge. We have a new plant. There are many, many types of plants that are built in such a way that they have pre-formed roots, and that makes propagation of those plants quite easy, like the philodendron hanging in your living room, or the monstera plant. Even though the leaves and the stems are quite thick, the procedures are the same. You can just cut a chunk off as long as it has the preformed roots, put them in a suitable potting soil, and they can root. So those are the easy ones. And you can see these are oftentimes aerial roots. So not only do they serve the purpose of rooting into the ground, they also can serve a structural function by, for example, if this vine, which happens to be a Swiss cheese monstera vine, this root can grow and attach itself to a tree, trunk, or a wall, or the side of your house, and it will produce kind of a glue and will help it stick to and provide some structure, some support for this vine so that it can grow vertically. But we take advantage of that little root because it also can root into the ground and provide the new root system for the cutting. And those are generally found at the nodes. That is the spot on the stem where the leaf comes out. It's called the node. The space in between the nodes is called the internode. So you can see there's a node with a root, an internode, another node with a root. So those are the simple ones. So the easiest cuttings. And here's an interesting scanning micrograph of an ivy. Ivy is another one of those plants that, because it loves to climb, it has preformed root. In this particular case, they're a preformed root, but sometimes all they have are a few cells. They're just sitting there dormant and waiting for the clue or the cue from their mother plant saying, oh, it's time to grow, you better grow because I don't have any other roots. But in this particular case, these little guys get up against the side of a tree or a wall or something, and then they grow and supply their glue, and that's why ivy can climb up your house. As I was saying, cuttings can go from easy to what some people might say is impossible, but I'd like to say really there's no way to really correctly say impossible, because a difficult cutting just means that we haven't figured out how to get it to root yet. Somewhere there's probably an answer, and we just need to keep looking until we find it. So in propagation, one of the most challenging things can be the development of the protocol to produce a particular type of plant. So how do we select cuttings? It really depends on the type of plant. If you have an annual plant, um, that it doesn't stick around very long. So it's not going to make a tree. It's not going to become woody. It's not going to have anything but nice fleshy tissue. If it's able to be propagated by cuttings, you basically would just cut a chunk off and you could probably root it under mist. But then when we get down to things like deciduous woody plants, you have branches and leaves in various stages all over the plant, and you have to decide how you're going to propagate that plant. Conifers are a special case. Most of them, they're not all, but many of them are evergreen. And so there are procedures to propagate each and every one of those, most of them. Herbaceous perennials, they kind of live in both worlds. They have uh, a perinating or a surviving portion of them that even during the dry or wintry times remains alive, whereas the herbaceous portion generally dies off. So you have the kind of the annual part and the perennial part. So that's what a herbaceous perennial is. And propagating it may involve the green herbaceous part, or it could involve the woody part. It just really depends on the type of plant. So besides the plant type, then in making cuttings, one would look at, okay, what is the age of the plant or its condition? Because sometimes small plants are the ones that are easy to propagate, and when they get older and more mature, it might become more difficult, or vice versa. Uh, there is a phenomenon called juvenility or maturity, which really doesn't have as much to do with chronological age as it does with animals, human beings, etc. It's a physiological condition. So juvenile parts of plants generally are more easy to propagate than the mature portions. And that's not 100%, but it's a general rule of thumb. In other words, when we look at all of these things uh, and we're going to propagate a new plant, we want to consider all of this basically to give us an idea of where to start, because a lot of times you don't have enough information to know which way to proceed. You want to use a general rule of thumb to start. So you have to, well, sometimes it takes a few go rounds before you really figure out how to propagate a certain plant. But you want to consider all of these factors first. So if a plant has juvenility or maturity, and an example of that is the ivy I mentioned earlier. It's like if you grow an ivy and it's growing on your house, and then later when you've had it on your house for years, you'll notice that certain branches stick out from the ivy and they're not clinging to the wall any longer. Those parts are generally what they call the mature growth, the only growth that you would see that would flower and set seeds, whereas the juvenile part continues to grow and cling to the structure. And generally in an ivy, that's the part you want to try to root, because if you make a cutting from the mature portion, you can make 100 cuttings. You might get one to root if you're lucky. Whereas the juvenile portions, you can cut and cut and cut, and every single one of those pieces will grow. So that's a very important phenomenon with some plants. Season of the year. Some cuttings should be made when a plant is freshly growing in the spring. Uh, so you want to take the fleshy portions and use those, whereas some other parts of the plant are used in other plants, like some fruit trees. You're not looking to make juvenile or fleshy cuttings. You might want to make woody cuttings after the leaves have fallen off the branches. And you cut those stems, and those can be aged in moist moss in a refrigerator or a cool area until spring. And then those woody stems can just be planted and they will grow in the spring. So that's an interesting phenomenon. Then we have cutting maturity. I'll give you a good example of that. You may or may not know a plant called the sweet almond, Aloysia virgata. It's become very popular here in north central Florida because it's so fragrant. I attempted to make cuttings of that for a few seasons because the first type of cutting we generally make are what they call the semi-hardwood cuttings. And those are not fully developed into hardwood, and they're not the fleshy brand new growth. They're somewhere in between. So what I found with the Aloysia plant is that these semi-hardwood cuttings were very difficult. And the answer finally was when I said, okay, let's try just the tips, like the tip 3 or 4 inches or the tip 5 inches of a freshly growing shoot. And lo and behold, those are the ones that rooted. So in that particular plant's case, we've solved a mystery. So in every new plant that we attempt, we want to find what the factors are and solve that mystery. And it can be quite fun. It can be challenging, and it also can be frustrating. So the softwood cuttings, there's no woody tissue at all. The semi-hardwood have started to color up, and they're usually thicker and a little harder because that woody tissue has started to form, but it's not hardened off yet. And then the hardwood cuttings, like one would do in the case of a fruit tree, etc., those are fully formed. They grew the full season, and then their leaves, in the case of deciduous trees, fell off, and those are used to propagate. Now, I said some cuttings are hard to do, but we do have something known as a rooting hormone. A rooting hormone is a chemical that basically it copies some of the signals that plants use hormones and produce hormones, just like animals and people have hormones, but they have different purposes. So plant hormones are little signals sent from one part of a plant to another to tell them to do things. So if a cutting is cut off a plant and it doesn't realize it like we realize, but the signals of the plant that come from a plant that has roots, not only is water going up there, but signals are coming from that plant going to the top of the plant saying, okay, we have got water, you can continue growing, or whatever the signal is. But those signals stop coming when you cut a cutting off a plant. So you have the base of that cutting, it has no roots, it has no source of moisture, so we have to keep it alive. Well, rooting hormones kind of fake the signals from the plant and give it the signal that it's time to root. And not all plants are alike, so there are many different strengths and types of rooting hormones that can be used, and some are more successful than others. So in the case of hormone, which is the one we preferentially use here because it comes in large quantities and it's not too expensive, there are different levels of this chemical indole 3-butyric acid, which happens to stimulate rooting in many, many cuttings. So you basically take the cutting, you may dip the end into one of these rooting hormones and then put it in a suitable media and try to keep it moist and keep the plant from dying while it's producing new roots. And the rooting hormone helps. Some of you may have seen Dip and Grow. That one has an additional chemical called naphthalene acetic acid. And in that case, because there's 2 kinds of chemical in that substance, if one doesn't work, the other one may. So get quite a bit of success using Dip and Grow, and this one is readily available even to any homeowner. You can buy it online, and it comes with a little mixing device and tells you just how to dilute the chemical in order to use it for certain plants, and that's a good starting point. Root tone is another one. Okay. So there's lots of different things. When you're trying to get cuttings to root, there's a few things you have to do. Reducing or removing leaves is important because when you've cut that cutting off, guess what leaves do? They transpire moisture. So if you leave all of the leaves on a cutting, the chances are they will wilt and the cutting will die because all of the moisture in that cutting will leave and no new moisture has come back in from the mother plant because it's no longer attached. So we reduce or remove leaves. We can increase humidity by putting it in a place where there's fog or mist. We can put the cutting in a moist environment that also provides oxygen. So let's go quickly through that, reducing or removing leaves. So this is a good way to start with a cutting because you've taken away half or more of what might remove all the moisture to that cutting. The humidity, well, commercially, we used a fog or a mist bench. I've heard of people doing that in their own places. If you're going to root lots and lots and lots of cuttings all the time, there are inexpensive ways to create your own mist bench or fog bench. So those are kind of cool. I've done it at home when I made a lot of cuttings. But otherwise, you can do things like surrounding your cuttings in a bag to hold in the moisture and humidity. You can buy little rooting structures that are little boxes that hold in moisture. Increasing humidity. So now the base of the cutting. So this is what I prefer to use for most of my cuttings. It's a mixture of two substances, perlite and vermiculite, and together they hold moisture, but they also provide aeration, which gives all the necessary oxygen for these plants to root. Vermiculite is what happens when you explode mica under high temperatures. So that same mica that Tiberius used for his greenhouse is now a rooting substance. Perlite is what happens when you take the beautiful natural glass called obsidian, volcanic glass, and you explode it at high temperatures like 2000 degrees. It turns into this popcorn-like structure and it holds a lot of moisture. So when you mix the vermiculite and the perlite together, you get an optimal amount of oxygen for your cuttings. Another thing we use is sphagnum moss. That's the one on the left. That's used for air layering, and well, hopefully I don't know if I'm going to have time, but we'll see. And then you have sphagnum peat moss, which is often used in potting mixtures. Two different things. Succulents have a different type of procedure to root those. You don't need all the fog and all the moisture. You need a little bit. Like you can root most succulents in sand. And the key to those is when you cut them off the plant, you let them dry off for a while so that a fresh cut does not get a rotting organism into it. So succulents are made to be able to survive and dry out. So what you do is you let the cutting dry out before you plant it, and you put it in a drier mix like a sand. And once in a while, you give it a little bit of water, and lo and behold, you will get a new plant. And when you use sand and rooting mixes, you can accommodate other types of plants that maybe aren't so picky as some succulents. Maybe there's a succulent plant like an epiphyllum that might need a little more moisture. Well, you can mix sand in with the other rooting substrates like vermiculite and perlite if you want to specialize it. Division, an easy one. You've probably all done it. You reduce the leaves because you're going to damage the roots when you pull the plant up and cut it into pieces. So that's why you see these cut back like this, and you cut your little plant into several pieces and plant them individually. Easy peasy. Common with irises, gingers. Gingers are very easy to produce this way. Many other plants that are monocots that clump. You could do it with bamboo, etc. Here's a ginger, just waiting to be pulled apart into separate plants. Got all of its roots too. Orchids were very commonly divided that way. This is a Cattleya orchid, kind of an ugly plant, but beautiful flowers. And you can see these are cut apart into little pieces, each one planted. And if you're lucky and don't rot them, a lot of times you treat the cut with fungicides so they don't get infected, and you can produce new orchids. Layering. Layering is an ancient art and science. Lots of different ways to layer them. And I mentioned earlier, this is how one makes cuttings without removing the cutting from the mother. How do we do that? A number of different ways. The simple layer, you just take the branch and you feed it down to the ground, pin it underneath the ground layer. And where you have pinned it down, that spot will generally root. After the season, you can check, dig that up a little bit. If it's got roots, you simply then sever this piece from the mother plant. Very simple, simple layering. Mound or stool layering, if there are many branches, but they're above the ground layer, you build the soil up around them. Kind of like when some people grow potatoes, how they build up soil around the potato plant and then potatoes are produced. Well, in this particular case, it might be an azalea or something that you want each of these branches to root. Well, you build the soil up around them and they root. It's called mound layering or stool layering. Then you can do the tip. You can bury the tip of the plant into the ground. You should pin it down, and that little tip will not only root, but then it will continue to grow and grows out of the ground. Then there's this crazy one that you do with vines, long vines. You can lead down into the ground, back up, down into the ground, back up. And you can do that as many times as you have vine. Pin them down. Every place you pin it down, roots, you cut them apart. Very simple. That's the serpentine or snake-like compound layering. Trench layering, that's a really cool one. I've not done it, but I'd like to. If you know you're going to layer this like this tree, you plant it at an angle so that it's closer to the ground to start with, and then you plant it in a trench, and then you gradually force that plant down to the ground. You pin it down, cover the trench, and then all these little buds will grow out of that plant, and each one of them becomes an individual plant that can be severed and planted. That's trench layering. Drop layering I've done, it's fun. It's very much like mound layering, except instead of this plant being in the ground, it's in a pot. Like I bought a big azalea bush from Lowe's, brought it to the greenhouse, put a second pot on top of the first pot, filled that with soil, and all the branches root. You can then cut those off, and you have, instead of one big azalea, you've now gotten enough to plant your yard. So that one's a good one, drop layering.
Dr. Schutzman, we need to wrap up.
Okay,
Thank
well,
you.
what I'll do, because I think we've gotten pretty good far enough. Grafting is something we could talk about at another time. But what I did want to bring up are some references. So one of the best references that is to become a member of the International Plant Propagators Society. And when you do that, you get access to about 75 years worth of their journal, which is packed full of different people publishing on the propagation methods they've used on many different plants over 75 years. That reference would fill shelves. It's so great. And also you'll be able to look back at these, because I'm sure this is going to be online. Here are some of the best references for plant prop. Okay,
Right. Thank you, Dr. Schutzman. Let's take a few questions.
sure.
What media do you use to start seeds?
Um, that generally it varies a little, but I get a good quality potting soil most of the time. Now that will do for most seeds, but if you want to grow things like succulents, then oftentimes you will use a fine sand for that, like a play sand. And that is the situation where you might put a heating pad underneath, some play sand, and you put the seeds on the surface because normally they're very small. And then you put a little scattering, like what you can use a salt shaker or a sugar shaker to shake some more sand over the top so that you're not burying them too deeply. And then you water them with a little fog nozzle. So you're very careful getting those little tiny succulents to start. When they come up, they're really, really tiny. And you get them to a certain size, and then you can use a small trowel and move them to separate pots. That would
Okay,
take the sandy soils. But for most things, I'll use a good quality potting mix. Something
great,
that drains. It can't be sopping wet. It has to have some aeration.
great, thank you. We have a few questions about rooting hormones.
Okay,
First one is, are rooting hormones safe to use for edible fruit and trees and vegetables?
absolutely.
Great.
Absolutely, because you're using and those chemicals are in almost every case they're um, they're created. I mean, they make the chemical, but the chemical is the same as what inherently is in that plant anyway. And then
Uh,
all you're really interested in is getting those roots to grow. That chemical does not travel after that, and you're putting
okay,
it very gingerly around the outside, and that eventually gets washed away, but it's there long enough to transmit the signal to the plant to produce roots. So it doesn't become a part of the plant.
okay. And what determines the strength of the rooting hormone that you
Well,
use?
there's some general guidelines that, I generally start any unknown cutting with a number 2 hormone because it's middle of the road. But if you don't get a good result, then you can go either of the 2 directions. And sometimes I'll go to the number 3 hormone because it's the strongest and see if that works. If 2 and 3 don't work, I'll go back to number 1. And just because one is weaker doesn't mean that it will always be less successful. Sometimes the weaker hormone works, but the higher ones do not. So each one of them is an individual thing that you have to test on its own.
Okay.
Okay.
Yeah. Let's see. Um, what is the best time of year to do air layering for camellia?
Oh, for camellias.
Or is there a better time than others to do any kind of
I think that
propagation?
I have seen air layering on camellias done when they are in active growth. And I believe, if I'm not mistaken, and of course we can always check on this, I believe that's following flowering. So you don't want to do it while they're in flower. You want to wait until that season passes and they start to go into active growth. And then you want to air layer, um, not the freshest new growth, but down below a little bit. So since layering oftentimes involves removing bark, they have to have some bark.
Sure. Okay, a couple more. Do you recommend software to track your plants? And if so, which one?
Software?
Yeah, do you?
No, I do everything the old-fashioned way.
Okay.
And what you can do, I mean, software you could keep a log, sure.
Mhm.
But I would recommend, if you're going to do a log, that you find a good kind of a journal that doesn't have an easily removable page, like one of those composition books, so
Okay,
that everything you write is there forever.
you're great. Any advice on transferring rooted cuttings into their containers?
That is a really good one. Of course, it's a very important feature because you can root and root all day long, but if you can't successfully transplant, then you're in a lot of trouble. Um, what I do, I try not to leave rooted cuttings too long together because then their roots start to intertwine. You don't want to have to rip them apart, otherwise you've destroyed a lot of work that you did. So uh, that involves sometimes exploratory investigation where you lift it up and see if anything very carefully with a trowel or something to see if there are any roots produced. Or you can jiggle the cutting just a little bit to see if it's still unattached, which means it has no roots yet. But if you feel that there's, it's holding, then you know that things have started, and you can experiment, lift up one and see what kind of roots you have. And then it's just a matter of uh, planting them so that the stem itself is not too deep in the soil. I like to put them where the roots start, just pretty much immediately under the soil level. And that might make the cutting a little wobbly. Well, in that case, you should stake it. Get some a fine bamboo stake, one of those the little wimpy ones, or a pencil, or whatever it is. Dig it down and tie it up. The most important part of that process is to not damage the roots, and then also to make sure that that cutting is stationary so that it can then grow.
Okay. Let's see. A lot of these methods and media are somewhat inorganic or synthetic in nature. Do you have any methods for fully organic propagation that you recommend?
Well, I have gotten out of the habit of trying to plant directly into soil media because if you try to propagate in the presence of every microorganism and fungus that's available in soil, you're going to defeat your purpose. You want, unless you have taken ground soil and sterilized it, you basically have to make sure nothing else is in there. Otherwise, you're bringing problems on yourself. So that's why we use you know, the vermiculite, the perlite, because they are very easily sterilized. They've been just by their very creation, they are sterile. You do
Great.
not want microorganisms and fungi are your enemy in propagation. So this is not the time to be talking about organic farming or any of that. The propagation has to follow as close to aseptic conditions as you can get. Now, once you've developed that into a plant, organic away. It's,
Great.
yeah.
Thank you. Let's see. One more question. How long should air layering stay on a citrus tree for a good take?
Well, I think most of them that work with air layering can produce roots within a month or 6 weeks. So when you're doing an air layer on a citrus, you want to have 2 layers of covering on there, because if you just do it with the clear plastic, you may be inhibiting some of that, and you'll be causing heat to dry out the media. That if you're wrapping it in sphagnum moss, which is traditional, uh, you don't want that to dry out. You want it to stay with a fairly good level of moisture. So you want to double wrap it. You want to put some heavy duty tin foil around the outside. And then once in a while, what you can always do, you basically have to investigate. You got to open up that aluminum foil and see whether you've got roots. Once you see some roots into that sphagnum moss, you're pretty much there. So the only thing you have to worry about is if you make a layer out of too big a branch, you won't have enough. If you make a layer and it's a standard size layer, but you make it on a big, huge branch, you won't have that layer won't be able to provide enough moisture to keep that branch alive. So you have to don't be greedy and make your layers on smaller branches that when those roots form, that those roots are sufficient to support the growth.
Okay. Let's see, I have one question that is asking if we will email this presentation. We will not, but we have recorded it and it will be available from our website,
Perfect.
floridafriendlylandscaping.com, under, I believe it's resources and then webinars. And it is a homeowner webinar. So Dr. Schutzman, thank you so much for joining us today. We really appreciate it. Great presentation. And we
Yeah,
will see,
sorry I couldn't give it as much attention to the last part of it. I'll have to,
we'll have to have you
first time
back.
around, it's always a little longer than I want it. And so
That's all right. We'll
we'll
have to have
tickle
you
it for
back.
the next time. But I hope that you all enjoyed what I did present, and good luck with your propagating. And if those of you who are interested in it should investigate the IPPS. They're a great organization and they have lots of resources.
Great. Thank you so much and happy holidays, everybody. We'll see you next year.
Thank
Thank
you.
you.